As I said then, in each one of the objectives that we're going to be looking at, we'll be clustering these organelles into groups. And the first group that we're going to look at are those that give the cell structural integrity. It's going to be the cytoskeleton and then something that's associated with the cytoskeleton called a centrosome. The structural integrity organelles that we're going to look at are the cytoskeleton and the centrosome. The cytoskeleton is just a word that means the skeleton of the cell. And just like your body has a skeleton, each cell has a skeleton as well. And just like your body gets its structural integrity from the skeleton, and the skeleton gives your body its basic shape, the cytoskeleton is going to give the cell its basic shape. Just as your body uses the skeleton as something to push or pull against, push against gravity, pull using muscles against gravity, then the cytoskeleton is going to be used to push or pull. And so there are cytoskeletal elements that are analogous to the muscles, and there are cytoskeletal elements that are analogous to the skeleton itself. So the cytoskeleton is going to be a very important organelle for the cell, as you might imagine. We're going to see several places where the cytoskeleton is very, very important. So in the cytoskeletal, we have these structural proteins, and we group them into three different sizes, and these three different sizes are made up of different proteins. The smallest of these, and this is not surprising when we look at the name, the smallest of these is a protein called actin, and actin, when it gathers together in strands, makes up a cytoskeletal element that's the smallest of the three, called microfilaments. Then in the middle category, called intermediate filaments, not too surprising, we have a whole bunch of different proteins which can make intermediate filaments in this intermediate category. They have names like keratin. We saw keratin earlier, recall, as one of the main components of hair, or as almost the exclusive component of hair. Vimentin is another one. Neurofilaments in neurons, as we might expect. Lamins, and there are many, many other proteins that fit into this intermediate filament category. We're not going to go through them all. We're just going to touch on a couple of the more common ones like keratin, vimentin, neurofilaments. And then the largest of the cytoskeletal elements are the microtubules. And they're made up of the protein tubulin plus some microtubule-associated proteins, also abbreviated MAPs or MAPs. A structure which has cytoskeleton at its core are what are called microvilli. In the intestines, there are villi, and the name means little fingers. So there are villi that are about 1 millimeter long. They're what give the intestines, if you look at the inside surface of the intestine, what we call the luminal surface, then they give that surface a velvety or shag carpet kind of appearance. And then even adding to that sort of velvety appearance are the microvilli, which are about 5 microns long. So they're about 1/200th the length of one of these individual villi. They're called microvilli. They're microscopic fingers. So we can see the villi, we can see things down to about 1/10 of a millimeter. So we can see the villi with the naked eye, but we can't see the microvilli with our naked eye. Each microvillus is the singular. Each microvillus has a microfilament core. And on the surface of the microvillus are these digestive enzymes that we've looked at earlier and cell flags that we've looked at earlier, forming a coat of sugar and protein called the glycocalyx. And all that means is a sugar coat. Glyco means sugar, calyx is coat or shell. So it's a sugar shell or sugar coat on the surface of these microvilli. Recall that when I was talking about electron microscopy, I said where you have cell membranes and where you have a lot of protein is where you have a lot of dark regions. And we can see that here. The cell membranes are the dark lines going up and around and forming the surface of each microvillus, up and down and up and down. We can see the cell membrane, the surface of the cell. These are being used to increase the surface area of the intestinal cell so there's more room for absorption. And then the heavy black stubble that looks like about a 2-day growth of beard on somebody who has very dark hair, that's the glycocalyx. And this glycocalyx also attracts a lot of those osmium atoms that have electron stopping power and so give this a black appearance. So the three categories of cytoskeletal proteins from smallest to largest, as we saw earlier, are the microfilaments. You can see them forming the core of a microvillus here. The intermediate filaments, such as keratin, vimentin, neurofilament protein, many other kinds of proteins. And then the microtubules, which are made up of tubulin plus microtubule-associated proteins. We want to look at a special organelle that's associated with microtubules, and that's the root of the microtubules, the place where they grow from, and these are called centrosomes. And a centrosome is an example of what we would now call a microtubule organizing center, or an MTOC, not to be confused with the MTC in Provo. That's a completely different deal. But these are microtubule organizing centers, or MTOCs. And they're the roots that the microtubules grow out of. Now, when we add together the centrioles plus the material that's around the centrioles, and the name we use for that is pericentriolar material, then we call that whole structure a centrosome. So a centrosome is an organelle which gives rise to the microtubules, which are part of the cytoskeleton. So we're considering it with these structural organelles. And these serve as a construction site for the microtubules. In Module 5, we'll look at the process of cell division in some detail. But right now what we need to know is that the cytoskeletal elements that we've been looking at, particularly the microtubules arising out of these centrioles, are an important part of cell division because they're the little tracks and motors that are going to move the different parts of the cells around as they begin to divide. One of the things that's going to be very important is a structure called the mitotic spindle. So in the process of mitosis or active cell division, there'll be one centriole in each of the developing cells. So we're going to get two daughter cells out of a single cell, and each one of those daughter cells is going to have its own centriole with its own microtubules growing out of it, forming this structure called the mitotic spindle. We'll look at this in more detail in Module 5. For now, we don't want to get confused by the names. So we have centrioles, centrosomes, and chromosomes, which are different combinations of centro and somo, or soma, all put together in different ways. And they're all in this somehow, but they're all used in slightly different ways. So we just want to make sure, especially as you go to take exam questions, that you know whether I'm talking about a centriole or a centrosome or a chromosome. So the chromosomes have centrosomes in the center of each one, and the centrosomes are where the microtubules that have grown out of the centriole are growing out and attaching to the chromosomes.